Hello, everyone. We are today with Rebecca Bunn from Western Washington University. And Rebecca is going to tell us about her recent perspective paper that she published in ISME called Revisiting the Direct Mineral Cycle Hypothesis, Arbuscular Mycorrhizal Fungi Colonize Leaf Litter and Why. Hello, Rebecca.
Hello, thanks for having me.
Thank you for being with us today. So can you explain us a little bit about your publication?
Sure. This paper is interesting because it actually started out as a suggestion in a very casual setting from David Janos to follow up on some of his students' research, which seemed to make logical sense because tropical forests are predominantly arbuscular mycorrhizal hosts. I live in the Pacific Northwest where we have a lot of ectomycorrhizal hosts. So we said, well, it'd be kind of interesting to see if this is happening outside of the tropics. So when we first talked, it was maybe 2011, and no one had yet looked anywhere outside of the tropics. And there were just a few papers documenting this phenomenon. So I went ahead and I got an undergraduate student who was interested, who went out and just took some leaf litter and started looking and said, yeah, there are structures that look like arbuscular mycorrhizal fungi. So let's see if we can do this in a little more robust way. So he went out and put out some leaf litter bags, and we did find colonization just using microscopic methods, which is a little difficult to prove because it's really hard to morphologically identify AM fungi separate from other types of fungi, especially when you're not in roots. So there's no arbuscules, and other things like vesicles are pretty rare.
Mm-hmm. So basically, hyphae.
Yeah, we're looking for hyphae. Yeah, and maybe spores that look consistent with AM fungi. So that was one really small undergraduate project. And then I had the next year another student come along who was really great, and I had been talking to David. We realized we need to have some molecular data to make this work. So the next undergrad, who's actually Dylan Simpson, who's the second author on this paper, did a really nice, much more complete study where he put out leaf litter in really fine bags. And he had a couple other things he was looking for. And with those samples, we were able to look both morphologically and do some extractions that focused on the small subunit. And again, this was me going to ILVA and saying, could you run these samples on the side? I really didn't have any funding for this work. It was all very sort of by the seat of our pants. So we were excited when we got those results that confirmed that that they were successful in amplifying AM fungi from these leaves. Also, we had some microscopic evidence, and we felt like this was a nice story and the numbers all correlate well. And then again, we were like, why? And so as I started going to the literature, yes, there were a couple that had documented AM fungi in leaf litter now from the Czech Republic. But all these pieces started coming together, like, why are AM fungi in leaf litter? Maybe it's actually not that surprising. So at that point, this became kind of more of a review of going back into the first observations from the tropics and up to today, where we're seeing some work coming out of Jen Jantz's lab and others really finding these mechanisms by which AM fungi maybe are cooperating with other soil organisms to access organic nutrients. So it was sort of the small study that when we started reading more about it, I feel like there's all these pieces coming together to explain why this might be happening. And that's how it turned into kind of a perspective with a small data set in it.
Mm-hmm. Yeah, so just to explain a little bit to all our audience, AM fungi, they don't have enzymes that can access nutrients from organic material, and this is the main difference with ectomycorrhizal fungi. So basically, when they transfer nutrient to the plant, it has to be already in a mineral form. So why do you think, how could they access the nutrient from the leaf litter? If this is the hypothesis that we have, that they are here to access these nutrients.
Yes. So backing up the direct mineral cycling hypothesis was this hypothesis from the mid 1900s that AM fungi were directly breaking down those organic compounds and accessing mineral nutrients that were organically bound. Today, as you said, we know AM fungi don't have the lytic enzymes to be able to access those organisms, or those, compounds. But this is what I was talking about, the work coming out of other labs looking at AM fungi trading labile carbon that's coming from plants with phosphate solubilizing bacteria. So trading carbon for phosphate in a very tightly coupled way. So it's not AM fungi by themselves, but maybe in cooperation with other decomposers. And the AM fungi may actually be stimulating the growth of those other decomposers by providing a pathway for plant carbon into the soil matrix. So there's a nice tight coupling maybe between AM fungi and phosphate, and for AM fungi and nitrogen, there might be a couple different steps from the decomposers to then maybe the grazers of the decomposers that are then releasing ammonium that AM fungi can take up. So that's the research that's coming out right now that kind of explains there may be a pathway there that's not the mechanism we were thinking of initially, but is functionally maybe the same, that we're transferring those nutrients fairly directly from organic materials like leaf litter to host plants.
The idea would be that the plant transfer the carbon to the mycorrhizal fungi, who will transfer to the bacteria, so the bacteria will do the mineralization and then up again. So I was wondering if there is already some evidence of this carbon all the way to the bacteria.
Yeah, so a pretty recent paper by Zhang and colleagues in the New Phytologist that came out very recently that was presented at ICOM in Merida, looking at that carbon and phosphorus exchange between the AM fungi and the bacteria in a very controlled system. So, you know, we're at sort of the baby step. How does that transfer up functionally? Is it the amount being transferred significant for the plants? And is it significant on an ecosystem level? And is it significant when we're growing in a matrix with lots of other organisms? Those are still questions that aren't answered.
Exactly. And so you have shown in your paper that the different studies that have been done before comes from there is a whole bunch of different ecosystems. And so do you think that this phenomenon is actually way more common than we think? Or is it something very peculiar in some places, depending on like the nutrient demand of the plants? Or what is your opinion about this?
So that's a good question. I think we haven't been looking. I think it probably is much more widespread than maybe we would believe from some of the predictions of Reed in the newer paper. You had the interview with Kabir about their paper showing the global patterns of AM and EM hosts. But I think that AM fungi are growing into leaf litter. It maybe is more common when mineral soils are really poor in nutrients, and so the main source of nutrients is in leaf litter. But it would be, and I think it also, we have to have an AM host plant there. So it follows the distribution of AM hosts, but they tend to grow in a lot of different places. And even when EM hosts are dominant, there's often as an understory that's AM dominant, or there are AM hosts within that forest ecosystem. So I think if people start looking, it's going to turn out not to be uncommon. And when you go back through the literature, that's part of our paper, we've been seeing AM fungi growing into all kinds of weird substrates so through the years. So I think it's just a question of looking. I do think there's still an open question of if that's an area AM fungi are actively exploring, like if they're sensing those nutrients and growing there because there's nutrients there. And also, what is the functional consequence on more of an ecosystem level are still pretty open questions.
Yeah. Well, I think it's a good invitation to everyone who's working with leaf litter. If you have samples and you're doing Illumina sequencing, please try to look for Glomeromycota. That would be actually really interesting.
Yeah.
Thank you very much, Rebecca. So are you still working on these questions? What are the next steps for your research right now?
Well, there's been a pause on this research. So after Dylan Simpson finished his work, I haven't had another student working on it. However, I think it's time to get another student out there. And I think the next questions might be trying to control access to the leaf litter, like maybe with cores in the soil, we could, and mesh and membrane, sort of exclude or allow AM fungi to access those leaf litter bags and start looking at differences in decomposition and yeah, I think that would be interesting to see how nutrients are moving out of leaf litter and whether it matters if AM fungi have access to them or not. I think that's the next question.
Yeah, that would be really good. Well, thank you very much for your time. And we will be back soon for more interview for the South American Mycorrhizal Research Network. Bye.
Thank you for having me. Bye.